Hey friends, welcome back to my channel. So let's start with types of viral nucleic acids. So viral nucleic acids can be DNA or RNA. In DNA, it can be single stranded with linear structure or circular structure, and double stranded DNA can be linear with single chain break or circular. For RNA viruses, single stranded and double stranded. Single stranded with linear positive strands. So these positive strands directly act as mRNA and negative strands are complementary to mRNA. So we have discussed all this in the previous lecture. Double stranded DNA, linear, double stranded and segmented RNA. Next, shape and symmetry of viruses. So all viruses possess nucleocapsid, that is nucleic acid plus protein. So symmetry refers to arrangement of capsomeres in capsid. First type of arrangement, helical symmetry. It's a rod shaped structure like this, arranged in a helix around the nucleic acid. So this internal part is the nucleic acid and these outer circular structure are capsid. Seen in filamentous viruses, example pleomorphic viruses or TMV, tobacco mosaic viruses. Next type of symmetry, icosahedral. So it is a spherical appearance, 20 triangular faces or 12 vertices like this, common in many animal viruses. Third type, complex symmetry, not purely helical or icosahedral. Example, T4 bacteriophage infects E. coli like this. Next is classification of viruses. So viruses are classified based on host type, virion structure and composition, mode of reproduction, disease cause, type of nucleic acid, DNA or RNA, single stranded or double stranded, presence or absence of envelope. So first one, host based classification. On the basis of host base, there are animal viruses, plant viruses, bacterial viruses, bacteriophages and insect viruses. So some common viruses, group and characteristics. So these are the virus families: Picornaviridae, Orthomyxoviridae, Parvoviridae, Adenoviridae and Poxviridae. So these are the genome sizes, type of nucleic acid they possess, strands, capsid symmetry, envelope and host. Bacteriophage, so bacterial viruses, they are the viruses that infect bacteria. So first observed by Twort in 1915 and Herrell in 1917. So they have given them the term bacteriophage, eaters of bacteria. Type of phages based on morphology, polyhedral, filamentous and complex. Complex phages have polyhedral head, tail structure, additional appendages, tail fiber and tail pin. We are talking about this structure. Capsid, collar, sheet, tail fibers and tail pin. So example of complete phage, that is T4 bacteriophage. Genome, linear double stranded DNA, size is around 172 kb. Structure, they have a head known as capsid. Shape is elongated icosahedral, contains viral DNA. Tail structure, collar, this structure connect head and tail. Sheath, contractile structure and core rigid in a tube. Then they have a base plate, hexagonal structure at the end, this base plate. And last, appendages, tail pin, 6 kinked tail fibers help in attachment to host. So these are the tail fibers. Phage genome type, most common double stranded DNA. Other genome type, single stranded DNA, example this double stranded DNA, T phages, lambda phages, single stranded positive strand, MS2, double stranded RNA, this. Classification of T phages, two group, T even phage, T2, T4, T6 and T odd phage, T1, T3, T5, T7. So, difference between T even and T odd, they differ in tail structure and chemical composition, very important. So, that is all for today. In the next lecture, we will start with life cycle of bacteriophage. Till then, bye.